Hi everyone, this is lecture 34 on the female reproductive system. So now we're going to continue from the previous lecture and show the physiological processes that can be quite complicated compared to the male reproductive system. And one of the major differences is that all the oogonia and mitotic divisions are completed prior to birth. As you saw in the male reproductive system, the spermatogonia can continually divide throughout the life of the individual. In the female reproductive system, they're all completed prior to. In addition, meiosis 1 of oogenesis begins in month 3 to 7 of fetal development. There, primary oocytes develop to prophase 1 and then stops. So as we see in this diagram, that prior to birth, the oogonia undergo mitosis to produce primary oocytes. And then it begins meiosis to undergoes meiosis from oogonia to produce primary oocytes that are then arrested in prophase 1. So we can see here that the oogonia and both primary oocytes are diploid in chromosomal number. During childhood, the primary oocytes remain in prophase 1 until puberty. So a girl has about 204 million in each ovary of a baby girl, many which degenerate. Puberty is down to about 80,000 to 400,000, and in her lifetime she may ovulate only about 400 to 500 secondary oocytes in her life. So here's what occurs from childhood to puberty, and you can see nothing happens here because they're all have been arrested in prophase 1. Now, from puberty to menopause, each month after puberty, 20 to 30 primary oocytes continue to develop. Now, this is important. It's not just one that develops, but there's a whole group that continually develop, and only one is selected to ovulate. Okay, so this will decrease the total numbers significantly over the lifetime. So what happens is one primary oocyte completes its first meiotic division to produce 2 unequal sized haploid cells. Okay, so this is significantly different from what we saw in spermatogenesis, where the cells are equal in size. But in oogenesis, we get one large cell and one small cell. The smaller cell is the first polar body, and the larger cell is the secondary oocyte. So we see here at puberty at the primary oocyte, which then continued to complete meiosis to produce a secondary oocyte, which stops in metaphase 2 each month. And so here the diploid primary oocyte now becomes a haploid secondary oocyte. And as we mentioned, the secondary oocyte continues to metaphase 2, and then development stops. Okay. The secondary here we see the secondary oocyte that's arrested in metaphase 2. Now, if the secondary oocyte is fertilized, meiosis 2 completes to form a second polar body and the fertilized ovum. Again, there's going to be a disparity in size. The ovum will be much larger, and the second polar body will be much smaller. So here in our diagram of oogenesis, we see the secondary oocyte producing the second polar body that really degenerates, and producing the fertilized ovum. But again, this only completes meiosis 2 if it becomes fertilized. So in the next figure are all steps of oogenesis. Okay, remember, prior to birth, the oogonia undergoes mitosis, and then begins meiosis to form a primary oocyte, which stops in prophase 1. So a girl has all the primary oocytes arrested in prophase 1 that she's born with. Then nothing happens between childhood to puberty. Then in puberty, the primary oocyte completes meiosis 1 to produce a secondary oocyte, and the first polar body is produced. Now the secondary oocyte is now haploid, and it continues to meiosis 2, but now becomes arrested at metaphase 2. It will only continue if it is fertilized by a sperm to complete meiosis 2, to produce a fertilized ovum. And if meiosis 2 is completed, a second polar body is produced, which then degenerates. So now in the next table, we're going to see a comparison between spermatogenesis and oogenesis. And there are significant differences between the two. So in the first column are different characteristics that the two are compared. Spermatogenesis is in the second column, and oogenesis is in the third column. So we can first look at the time of onset. Spermatogenesis begins at puberty, whereas oogenesis begins prior to birth. Next, what occurs at puberty? In spermatogenesis, it begins and continually produces sperm cells. In oogenesis, it continues meiosis and releases an immature gamete, the secondary oocyte, each month. The number of cells that are produced between the two. Spermatogenesis produces millions of small motile sperm each day, whereas oogenesis produces one large oocyte each month. When the processes end, well, spermatogenesis continue until death, whereas oogenesis generally stops at ages 45 to 55. The duration of meiosis in spermatogenesis continues throughout life, whereas oogenesis has two pauses. One at prophase 1 from prenatal development to puberty, and then again at metaphase 2 from ovulation to fertilization. So we see here there's a significant differences between spermatogenesis and oogenesis. Now, what makes things more complicated is that in oogenesis, not only does the cell mature, but also the follicles surrounding that cell matures. Okay. So we have follicular development, and this is another difference between spermatogenesis and oogenesis. So the oocyte grows within a follicle in the ovarian cortex. The first ones that form are called primordial follicles. And so the primordial follicles are the first follicles formed in the female fetus. Each one contains a primary oocyte. So here now we're looking at the second column of our development. And we see the primary follicles surrounded by follicular cells with the primary oocyte in the center. Next we have primary follicles. These develop from some primordial follicles as the primary oocytes enlarge during childhood to puberty. Many die by apoptosis, decreasing the total number by 50 to 90%. So this is a significant drop. Apoptosis is programmed cell death. So some are programmed to die. So we see here in our development of follicle that primary oocyte now matures in a primary follicle that has to differentiate to granulosa cells. Okay. Next, we have secondary follicles, and these develop from primary follicles with each containing an arrested primary oocyte. So here's a secondary follicle. Now the granulosa cells have differentiated into theca cells, which we'll describe later. And you can see pools of follicular fluid are forming, and eventually they're going to fuse. So we have a primary oocyte in a primordial follicle. We have a primary oocyte in a primary follicle, and we also have a primary oocyte in a secondary follicle. Finally, we have a vesicular or tertiary follicle. And about once a month, one matures from a secondary follicle. It contains a secondary oocyte stopped. Remember, it contains a secondary oocyte stopped at metaphase 2. Okay, and we can see that the follicular fluid pools have merged to form one large pool. This is very characteristic of a vesicular follicle, and it still contains a secondary follicle, a secondary oocyte. Each month, a secondary oocyte will ovulate and will rupture, resulting in the ovulation of a secondary oocyte. Okay, and of course, at menopause, the follicle stops developing ova. Okay, so here we show a vesicular follicle undergoing ovulation, and we see it's bursting, producing the secondary oocyte, and it retains some cells follicular cells surrounding it, which we'll describe later. Okay, so we have a summary of follicular development, where we have a primordial follicle maturing to a primary follicle, which matures to a secondary follicle, to a vesicular follicle. Okay, each containing a primary oocyte and a secondary oocyte in the vesicular follicle. When it ovulates, the secondary oocyte leaves and bursts from the surface of the ovary, leaving a structure that will produce a structure called the Corpus Luteum, which we'll describe in a little bit. But notice that the complexities involved in oogenesis is that not only is there several pauses and a disparate size in the cells that are produced, but also changes in the follicles surrounding those oocytes. It makes it a little more complicated. Now, to add a little more, another level of complexity, the female reproductive system has what's called the Ovarian Cycle, and these are the monthly events associated with oocyte and follicle maturation. Okay, so in reality, an oocyte requires more than 350 days to go from the primordial follicle to ovulation. It's that they're all staggered and maturing at different stages. So the ovary has follicles in various stages of development. The ovarian cycle is divided into the follicular phase, ovulation phase, and the luteal phase. Okay, so let's look at the follicular phase first. The follicular phase is broken up into 1 to 4 stages, and during this time the follicle grows and develops further. The follicular phase is divided into these 4 stages. We have a primordial follicle, which we described already, a primary follicle, which we described already, and a secondary follicle, and a tertiary or vesicular follicle. So essentially what we described earlier was the follicular phase. So let's describe in a little more detail of each of these. In a primordial follicle, each primary oocyte is surrounded by a layer of squamous follicular cells. At birth, each ovary has about 204 million containing a primary oocyte. So there are many of these primordial follicles at birth. And during childhood, they begin developing into primary follicles. So here we see a primordial follicle, each containing a primary oocyte surrounded by follicular cells. And these follicular cells are flat cells called squamous cells, a single layer of squamous cells. Okay. These form into a primary follicle. Okay. And these follicular cells then become more cuboidal granulosa cells. And in these granulosa cells, microvilli grow, forming a structure called the zona pellucida. Okay. The microvilli will increase the surface area for the transfer of materials through gap junctions. In addition, thecal cells will develop, but only a small number of these follicular cells continue to develop. So here we have the primary follicle, okay, still with a primary oocyte in it. The granulosa cells have differentiated into thecal cells. They have transformed more into cuboidal granulosa cells. and in addition to microvilli, and as we mentioned, thecal cells develop. The third stage is the secondary follicle. And here the follicle wall thickens, and as we mentioned, the granular cells start secreting follicular fluid. These thecal cells begin to produce estrogen, and generally one secondary follicle per cycle will complete development, which will then ovulate. So here's a secondary follicle. Okay, you can see the pools of follicular fluid. Okay, and the granulosa cells. But now we have multiple layers, and of course the small pools of follicular fluid. Finally, we have a vesicular or tertiary follicle. This is where the pockets of the follicular fluid merge to form a cavity called an antrum. So the antrum is the now pooled follicular fluid. The primary oocyte and granular cells is known as a cumulus, and they project into the antrum on a stalk. Okay. So here's the vesicular follicle. And so here's the antrum. Here are the thecal cells, the secondary oocyte, and the granulose cells surrounding the secondary oocyte. Okay. And so at this point, the primary oocyte finished meiosis 1 to become the secondary oocyte, and surrounded by granular cells and fluid-filled antrum. And as we mentioned, to get to this stage takes more than 350 days. Now the last stage is the vesicular or tertiary follicle, and this enlarges to 15 to 25 millimeters in diameter. The primary oocytes will then complete meiosis 1, forming the secondary oocyte and first polar body, and then stop at metaphase 2 unless fertilization occurs. So here's the vesicular follicle. Okay. Here's the large antrum and the granulosa surrounding the secondary oocyte. The second stage of the follicular phase is the ovulation stage. This is when the ovary expels the secondary oocyte and its associated granulosa cells. So the ovary wall actually ruptures to allow the follicle to expel the secondary oocyte. Now, the secondary oocyte is still surrounded by its granulosa cells, but at this time after ovulation, now it's called the corona radiata. Okay. So the corona radiata is surrounding the secondary oocyte after ovulation, and the vesicular follicle then collapses. So here we see the ruptured follicle. Here's the secondary oocyte that's ovulated, surrounded by the corona radiata. Finally, the third stage is the luteal stage. And the ruptured follicle is still retained and becomes an endocrine organ called the corpus luteum. Okay. And the corpus luteum will produce progesterone and estrogen for 3 months to maintain the pregnancy if fertilization occurs. Okay. If no fertilization occurs, then it'll degenerate in 10 days. And so this will serve to maintain the pregnancy until other structures are formed to take over. So here's the corpus luteum that produces becomes an endocrine organ to produce progesterone and estrogen. Finally, if fertilization does not occur, macrophages invade the corpus luteum and fibroblasts lay down collagen, and so it remains as scar tissue on the ovary and becomes a corpus albicans. So this will conclude one ovarian cycle. So here we show the corpus albicans, which is scar tissue. And of course, it will stop producing hormones because fertilization did not occur. Okay, so now we look at the completed ovarian cycle. Okay, and of course the textbook puts all the stages in subsequent numbers, even if they're in different stages. Okay. Now we're going to talk about the hormonal interactions during the ovarian cycle. Okay, and so the average ovarian cycle is about 28 days and requires hormonal changes from the hypothalamus, anterior pituitary, and the ovaries. Okay, and this is the similar figure which we showed in the endocrine system. We have the first tier control, which is the hypothalamus releasing the gonadotropin releasing hormone. Okay. So here we have the first tier control. The hypothalamus releases gonadotropin releasing hormone. It secretes it to the anterior pituitary, which releases luteinizing hormone and follicle stimulating hormone in response to the GnRH. In response, the ovaries secrete androgens in response to luteinizing hormone. And the ovaries has the ability to transform the androgens into estrogens and then secrete the estrogens as well as inhibin in response to follicle stimulating hormone. The effects of these hormones: the estrogens stimulate a dominant follicle to mature to a vesicular follicle. Okay. And the new vesicular follicle itself can produce large amounts of estrogens, which triggers an LH surge. The LH surge and FSH triggers ovulation. So normally, estrogens will inhibit LH secretion at the level of anterior pituitary, while estrogens and inhibin will decrease follicle stimulating hormone, and estrogens will inhibit gonadotropin releasing hormone. So this negative feedback is nothing different than we've experienced before with the endocrine system. However, in the female reproductive system, something unique happens, is that the vesicular follicle produces more estrogens. This is this dominant follicle that will ovulate, and this will cause a positive feedback toward the anterior pituitary, which will produce large amounts of surge of LH as well as FSH, and this will induce or trigger ovulation. So what's unique in the female reproductive system is this this positive feedback to cause the LH surge to stimulate ovulation. Okay. If fertilization occurs, progesterone from the corpus luteum will inhibit further release of gonadotropins. So this will prevent the maturation of other follicles in case fertilization occurs. If fertilization does not occur, the corpus luteum will degenerate into corpus albicans, as we mentioned before, and gonadotropin releasing hormone will rise, and the cycle begins again, because once it degenerates, it will stop. It won't be producing progesterone anymore, and so the inhibition on the gonadotropins from the hypothalamus will not be present. Okay. There are also other effects of female hormones. Okay, estrogen and to a lesser extent progesterone will stimulate the female sex characteristics. Okay, so here are some of the effects of female hormones. It will maturate the sex organs. It will help develop the external genitalia. It will produce breast development and also fat accumulation. It will decrease osteoclast activity. It also will increase the healthy high density lipoproteins and decrease the low density lipoproteins. So these are some of the effects of female hormones. Okay. So now, in addition to the ovarian cycle, we also have what's called the uterine cycle or menstrual cycle, because the uterine lining has to prepare the uterus in case fertilization does occur. And so this is the cyclical changes that occurs in the uterine endometrium. Okay. Now, if you recall, the endometrium has two main layers. It has an outer superficial stratum functionalis and an inner deeper stratum basalis. Okay, and then of course, deeper to this is the lumen of the uterus. So here I made you a table to describe the two layers, the stratum functionalis and the stratum basalis. Now, the cyclical changes that we see occur in the uterine cycle has mainly to do with the stratum functionalis in response to ovarian hormones. And so it will thicken and develop throughout the uterine cycle. However, the stratum basalis does not thicken in response to ovarian hormones. It stays the same. If fertilization does not occur, this thickened lining will be shed as 35 to 50 milliliters of blood discharge from the vagina once per month during menstruation. And then is the stratum basalis will form a new stratum functionalis after menstruation ends. So even though the basalis does not change during the uterine cycle, it's responsible for producing the new stratum functionalis for the subsequent monthly cycle. Okay. So in this diagram, first we see this inset here, and we see the two layers. We see the stratum basalis here, and the stratum functionalis here. Okay. And so here we see the relative thickness throughout the uterine cycle between the stratum basalis, okay, which doesn't change too much. But here, the stratum functionalis, we see significant changes in the thickness and the vascularization of the functionalis. So let's go look at this in a little more detail. So the uterine or menstrual cycle, we have what is called endometrial or uterine glands that lengthen as the endometrium thickens. And so blood's supplied through the spinal arteries and veins. Okay, so here we have the endometrial glands that are lengthening, and we also have the lengthening of the spinal arteries and veins that are going to maintain the vascularization of the stratum functionalis as it thickens. We can see it thickening over the 28-day cycle. Now, the uterine cycle occurs in 3 phases, and these are the order that they experience it from on the 28-day cycle. We have the menstrual phase first, okay, and then we have the proliferative pre-ovulatory phase, and the secretory post-ovulatory phase. Okay. So the menstrual phase occurs on days 1 through 5 of the menstrual phase. It actually began earlier from the previous cycle. So this is the shedding of the stratum functionalis during menstruation from the previous monthly cycle. So we see in days 1 through 5 the decrease in thickening as the stratum functionalis is lost. Okay, and resulting in menstruation. We can see that it actually started to begin on day 28 from the previous cycle. The second stage of the uterine or menstrual cycle is the proliferative pre-ovulatory phase. This occurs over days 6 through 14. In this case, the new stratum functionalis develops with spinal arteries and veins. The layer thickens, endometrial glands enlarges, and the spinal arteries and veins increase in number. Okay, and in addition, thick and sticky cervical mucus thins prior to ovulation allows sperm passage to the uterus. And ovulation will occur on day 14 at the end of the proliferative phase. So here we see that in the proliferative phase, the stratum functionalis develops endometrial glands and small artery veins and thickens as it progresses through day 6 to 14. And day 14 is when ovulation occurs. Finally, the third stage of the uterine or menstrual cycle is the secretory or post-ovulatory phase, which occurs during days 15 to 28. And the spinal arteries convert the stratum functionalis into a secretory mucosa. The endometrial glands secrete glycogen-rich fluid called uterine milk to sustain the embryo and fertilization occurs. So if pregnancy does not occur, cells of the stratum functionalis die and the menstrual phase begins on day 28. If pregnancy does occur, the secretory phase persists and the uterus continues to develop to prepare for implantation of the fertilized egg. Okay, so during the secretory phase, the small arteries convert the stratum functionalis into the secretory mucosa, and the endometrial cells start secreting the uterine milk. However, if fertilization does not occur, on day 28 the menstrual phase begins, which continues into the beginning of the next monthly cycle. Okay. Now, in addition to this, the hormones fluctuate during the uterine cycle to control the changes that we find. So the rhythmic fluctuation of hormones from the hypothalamus, pituitary, and ovaries help control the uterine cycle. So let's look at the beginning. At the start of the menstrual phase, both FSH and LH levels rise. The ovarian hormones are lowest until day 5, when the follicular cells start producing estrogen. Okay, so we see here that LH and FSH begin to rise very early. Okay. And the ovarian hormones are low at the beginning of the menstrual phase. Estrogen production rises around day 5 as the follicular cells start producing estrogen and progesterone. And it continues to rise throughout the proliferative phase. The rise in estrogen in the proliferative phase causes the stratum functionalis to make progesterone receptors. Okay, so they'll be more sensitive to the production of progesterone. Estrogen triggers the release of more FSH and LH, which peaks at day 14 to cause ovulation. Okay, so here we see the increase in estrogen. Okay, progesterone levels rise after ovulation due to the formation of the corpus luteum, and this rise in estrogen will stimulate the LH and FSH surging, which is due to the positive feedback, which will stimulate ovulation on day 14. Now, the progesterone from the corpus luteum in the secretory phase increases to inhibit the anterior pituitary, so that further maturation of follicles are prevented. And this will lead to low FSH and LH levels, and as a result, estrogen also decreases. So LH and FSH decrease due to negative feedback on the anterior pituitary gland, and the progesterone levels rise throughout the secretory phase, but drop around day 24 if fertilization does not occur. Okay, but if fertilization does occur, then it will continue. So these are the major changes in the hormone levels during the uterine cycle. Now, if it's not fertilized, we mentioned that corpus luteum will degenerate at the end of the secretory phase. As a result, its function as an endocrine organ will stop, progesterone decreases, and the stratum functionalis will break down to start the menstrual phase. Okay, so we see the endometrial lining functionalis will diminish, but then regrow on each monthly cycle. So here we have, in this figure, we have the gonadotropic hormone levels change, the ovarian cycle hormones, and we have the endometrial lining development, and finally also the ovarian cycle, all in the same figure. So we can see how everything occurs on the same 28-day cycle. So for example, we have high levels of estrogen produced to cause the stimulated LH, and to a smaller degree FSH to cause ovulation. Okay. Now, if fertilization does occur, the outer layer of the embryo starts secreting a hormone called the human chorionic gonadotropin, or HCG. HCG is important because it keeps the corpus luteum from degenerating, and it also maintains progesterone levels high. Okay, and since this is HCG is a is a specific sign for fertilization. It's also the target for home pregnancy tests. Okay. Now, the female sexual response is similar to males. The vaginal mucosa, vestibule and breast engorged with blood, the clitoris and the nipples become erect, and the vaginal mucosa and vestibular gland secretions lubricate the vestibule. Okay. However, differences that do occur is that there's no ejaculation or a refractive period, as in males. Okay. Peristaltic contractions of the uterus and cervix pushes down to assist in moving the semen deeper into the vagina. Okay. At puberty, the hypothalamus is less sensitive to the inhibition to estrogen and progesterone for ovaries. So this is why at puberty, these significant cyclical changes begin to occur. It begins around ages 9 to 11 as breast development begins and female secondary characteristics develop. Female secondary characteristics include pubic and ancillary hair, increase in adipose tissue, adipose deposits in the breast, hips, and thighs, an increase in height, increase in sebaceous gland secretions, and the widening of the pelvis. So these are the general female secondary characteristics at puberty. In addition, about 2 years after puberty is menarche, which is the first menstrual bleeding. Okay. Interestingly, this menarche does not occur if body fat is lower than 15 to 27%. So those females that maybe have eating disorders, and they might have too low body fat, menarche will not continue because the body doesn't have body fat to support a pregnancy. And the way the body interprets this is through a hormone called leptin, which is secreted by the adipocytes that stimulates gonadotropin secretion. So body fat will influence menarche. Okay, before we continue on, we'll look at the effects of aging and a period called menopause. It's where the menstruation stops for more than 1 year due to age. Okay, this occurs about the late 40s or 50s, where menstruation becomes increasingly irregular, and it will eventually stop. There's a period of what's called a female climacteric, in which the time between the irregular menstrual cycles and menopause occur. So female climacteric precedes menopause. Okay, and it occurs because of the low number of primary follicles that respond to FSH and LH. They just run out of viable primary follicles. And this occurs due to a process called atresia. And this is the degeneration and reabsorption of the immature follicles. Okay, so let's look at the major reproductive hormones that are involved in this table of all the hormones that we discuss in this lecture. Okay, so this includes the female reproductive system, and in the next lecture we're going to talk about fertilization and embryogenesis once fertilization has occurred. Okay, and so the next lecture is a little bit longer because it includes a lot of different material. So this concludes lecture 34 on the female reproductive system.